have interaction with the stakeholders in Pakistan and globally. Some participants are also there, so we will have a good discussion and good outcomes of these seminars. So today's seminars basically have a theme of global wheat yield enhancement strategies. So we will listen our speakers on this aspect with a different point of view. So here we have three experts. One of them is Dr. Ravi Singh, who is the head of wheat breeding at CIMMYT, Professor Vara Parsad, who is director of sustainable wheat production innovation lab, and Philomin Juliana is quantitative geneticist in the Borlaug Institute. So these speakers will talk with us. I just see if Professor Dr. Ikrar Ahmed Khan has joined. He was traveling. If he has joined, I will request him to few words for the participants and this webinars. Sir, are you there? Asad?
No, sir, I cannot see him in the participant list. So I think at the moment he is not with us. But I will keep looking and I will inform you once he is with us in this webinar. And I suggest we should move forward.
So we will move to our first speaker, Dr. Ravi Singh. He is head wheat improvement global wheat program of International Maize and Wheat Improvement Center, Mexico. I personally met Ravi Singh in 2008 at PBI in Australia. Once I was working with Richard and Harbans who are there. So after a long time, I will listen Ravi Singh in this webinar. So over to you, Dr. Ravi Singh.
Okay, good afternoon there. Let me see if I could first share the screen here. I've been using Zoom, you know. So
Most people use Zoom. So
no, I just need to see the share the screen. How does it work here?
It's on top and right corner.
And
next
the
to the camera and the mic.
where red leave button is there, and on the left there is an arrow written there, share. You just click on that.
Yeah, somehow, you know,
on the top right.
Here is people, chat, reaction, more, camera.
Oh, yeah, my camera, there should be a share, a small arrow, and there will be written share.
Yeah, it's just somehow, you know, the screen has become all of a sudden small, you know. And it's not moving, looks like frozen.
Sir, use Control Shift M. Use Control Shift E key, a shortcut key. Control Shift E, and you will get the sharing.
Dr. Ravi, we can see your screen. So under Share, there's an option called Windows. If we can now see your screen, but the team screen is seen. So there's an option called Windows under Share.
Philomin, my screen froze, you know. So maybe I need to get out and restart. I'm sorry about that, but something happened. It's only part is showing on my. Oh, okay, now okay, share is moved. And I had put my talk here. This is interesting. It's
the Windows button, we have to double click. Windows.
yeah. Okay, start now. So what do I do now? Share.
Double click on the windows and then it shows all the open windows and their PowerPoint is there.
Where is the, so here, window.
Double click on the window and then it takes you to the PowerPoint.
And double click.
Yeah, but sir, first make sure the PowerPoint is already open on your computer.
Yeah, PowerPoint is open, that's the thing.
Or if you would like, can you email me your PowerPoint? I mean, I can play from here.
Maybe that could be one way.
Yes, sure.
Interesting. Should have tried it out before. Maybe you could start with Dr. Vara Parsad in the meantime, if you wish, so we could figure it out in the meantime.
Okay, okay. We proceed with Professor Vara Parsad. He is Director Sustainable Intensification Innovation Lab, Kansas State University, USA. So over to you, Professor Vara Parsad.
Yes, Dr. Singh, if you can stop sharing your screen, I will start mine.
So my
yeah, it's visible, Vara Parsad. It's all right. Yeah.
Okay, you can see the full screen or not yet, I believe. So you're seeing the full screen now, right?
Yeah, full screen. Yeah.
All right. Yes. Good evening to all of you. Hopefully you are able to hear and see the presentation nicely. First of all, I would like to thank Dr. Ehsan Khan for inviting me to this webinar series being organized by the University of Agriculture, Faisalabad, Pakistan. And thank you to Dr. Zulfikar Ali for moderating this session. And today's I'll be talking about the impact of high temperature stress on wheat, the complete story. I will provide a summary of most of the items which we'll be facing under the threat of climate change. And at the beginning, Dr. Khan and Dr. Ali, both of them mentioned the importance of climate change and how it is impacting the global food production systems, particularly the heat wave, which has been happening in the Indian subcontinent, including Pakistan and other countries, and how it can potentially impact the wheat productivity. And in addition to that, we have an issue of Russia and Ukraine conflict, which is also impacting the exports from the troubled regions to the rest of the world, especially Asia and Africa, and it will have significant impact on the availability of wheat. So today I'll be this will be the outline of my presentation. In the next 30 minutes, 25 minutes, I will introduce you to a little bit about climate change and temperature. What are the recent trends and how does the future look like in terms of high temperature stress? And then I'll specifically answer 7 of these questions, which are most often asked to the wheat researchers. Number 1 is, what is the most sensitive stage to high temperature stress? Number 2, which has greater impact and stress? Is it the daytime temperatures or the nighttime high temperatures? Number 3, what are the key physiological processes that are impacted and what are the key reasons behind that? Number 4, do high temperature stress have similar effects at different growth stages? Number 5, what are the reasons for lower seed numbers and seed size under stress conditions, particularly heat stress? And is there a genetic variability for key traits associated with high temperature stress? And the last question is, how do wheat compare to other grain crops in terms of their temperature response? And then I'll conclude and have a little remarks of what the future direction of wheat research should be. So let me start with climate change and climate variability, recent changes and how the future scenarios looks in terms of temperature stress. We all know that the greenhouse gas emissions have been significantly increasing, particularly the four which I've shown here, the carbon dioxide concentrations, the methane emissions, the nitrous oxide emissions, and the sulfur hexafluorides. All of these four are extremely potent greenhouse gases, which has the ability to increase the global temperatures. At present, last year, 2021, the average carbon dioxide concentration across the world was 416 ppm, which has been the highest so far. Just for relevance, in 1960s, it was less than 320. So we have significantly increased the carbon dioxide concentration, and same is the case with methane, nitrous oxide, and sulfur hexafluorides. And what it has done to the global average surface temperatures, if you look at this visual, this shows you the global temperatures from 1850 to 2020. The mean temperatures have increased anywhere between close to 1 degree Celsius, particularly in 2020 and 20 parts of 2021. The temperatures were close to 1.2 degrees Celsius warmer than what it was in the average of 1850 to 1900. So that's a significant shift, and these will have negative impacts on productivity of grain crops, particularly if they occur at sensitive stages of crop development. And in future, we might not only see the increase in the average temperatures, but also we'll most likely see occurrence of extreme events, short periods of extreme events, whether it is extremely hot weather or on the cold side as well, extreme cold weather events. We are already seeing it right now. I was reading the news and they said that Indian subcontinent has recorded the highest average April temperatures in the last 120 years. So that's a significant shift, and this is occurring at a stage when it might have an impact on productivity of grain crops. And similar things are also happening in the northern hemisphere, particularly including in US and Kansas. We are seeing extreme hot events, which will have negative impacts on our food productivity, especially on wheat as well. So I'll now go ahead and answer those 7 important questions. And for each of these visuals, which I'm going to show, there is a reference. Most of these are published literature from my group. And if you would like to know a little bit more details about methodology, please reach out or go ahead and refer to that original paper. I'm not going to go too much in detail considering the time we have to discuss today, but I'll be open for questions at the end of this if there is time. So the number 1 question, what is the most sensitive stages to high temperature stress? We all know that all stages are not equally sensitive. Some of them are more sensitive. And in general, reproductive stages of crop development are relatively more sensitive to heat stress compared to the vegetative growth. And not only that, but within the reproductive period, there are certain times where the sensitivity is even greater, because when we say reproductive phase, it starts from the growing point differentiation all the way through the gametogenesis, flowering, seed set, and the seed development stage all the way through maturity. So it's a very large long duration, almost like 40-day period. But within that, there are certain stages which are even more sensitive to heat stress. So in this visual, and your x-axis is the time of temperature stress relative to flowering. 0 is flowering, and -5, -10, -15 are days before flowering. And positive signs 5, 10, 15, 20 are the days after flowering. And I'm showing you the speculate fertility data as a percentage of control. As a percentage. So control and control is where the plants were exposed to optimum temperature, 25 daytime high and 15 nighttime temperatures. And the high temperature stress was 36 by 26, and the duration of stress was 5 days. So as you can see, the maximum reduction in spikelet fertility is happening at flowering, 0, and few days before flowering, 5 to 10 days before flowering. Those are the two most sensitive stages when the spikelet fertility is the lowest. Whereas after 5 days, the effect on spikelet fertility is not too much. So those two stages, at the time of flowering and 5 to 10 days before flowering, is the most sensitive stage with respect to its impact on spikelet fertility. Whereas if you look at the figure on the right-hand side, when the stress is happening 5 to 10 days, especially 10 days after flowering, that's when you see the maximum reduction in the individual grain weight or the seed size. So that's where the rapid grain filling is occurring, and that's when you see the negative impacts of this 5-day heat stress period. So what are the things which are happening at 5 to 10 days before flowering? The viability of the gametes, the pollen viability, the ovule viability is being determined. So that's the one which is impacted. Whereas at 5 to 10 days after flowering or seed set, that's when the maximum grain filling is occurring. So those are the two reasons why you see a reduction in the spikelet fertility and the individual grain weight. And we actually went ahead and pinpointed even to more precision by exposing the plants to 2 days of high temperature stress. And again, now it's very clear the 2 days before anthesis and 8 days before anthesis, that's a time frame which is extremely sensitive, even to 2 days of high temperature stress. And it impacts the pollen viability, which is the live pollen grain percentage, which is being shown on the figure on your right. So those are the two things which is being impacted: the spikelet fertility because of the loss of pollen viability when the stress is occurring 2 to 8 days before anthesis. So those are the most sensitive stages. Now the second question is which has greater impact stress, daytime temperature or nighttime temperatures? We know, those of you who have read the IPCC reports, there's been clear evidence that it's the nighttime temperature which is increasing at a greater rate compared to the daytime temperatures, and these will have significant impact on the productivity of the crops, not only in terms of their impact on respiration, but also in terms of affecting other physiological traits which are important for yield formation and growth of seeds. So we did multiple experiments again to look at the impact of optimum temperature, high nighttime, high daytime, and the combination of high day and night. In this visuals, the controls are red, and then the high nighttime temperature is blue, and the high daytime temperature is orange, and the combination of those two, high day and nighttime temperature. Again, I'm showing you the data on spikelet fertility and the grain numbers. As you can see, high nighttime temperature significantly decreased the spikelet fertility and ultimately led to the lower grain numbers. However, the combination of both high day and nighttime temperature had significantly greater effect on both spikelet fertility and the grain numbers. So in this case, what we are seeing is the effect of high day and high night at that intensity both had equal effects on spikelet fertility, especially on which is resulting in the lower grain numbers. But however, on the figure on your right shows that high daytime had slightly greater effect on the grain numbers compared to that of the high nighttime temperatures. Again, the combination of those two day and nighttime temperatures had significantly greater impact compared to individual stresses. And not only that, if these temperatures were happening at the time of anthesis, the primary reasons, in addition to the spikelet fertility, the other things which was being impacted was the leaf photosynthesis, the membrane damage, and the primary reasons for loss of photosynthesis was the membrane damage. And then the reasons for membrane damage was the increased reactive oxygen species level and decreased antioxidant capacity. So those were the physiological traits which are impacting the wheat, and that therefore you saw the decreased photosynthesis due to membrane damage, increased reactive oxygen species, and lower antioxidant capacity. And that is true for both vegetative and the reproductive membranes. And therefore, you saw previously the loss of pollen viability and the decreased seed set. So the number 3 question is, what are the key physiological processes that are impacted and the reasons under high daytime or high nighttime temperatures? I've already showed you and answered some of those questions in the previous figure, but this is a more comprehensive and the detailed information here. In this visuals, you have most of the physiological traits that seed set, which have direct impact on the yield component: seed set, grain number, individual grain weight, grain yield per plant, and also per spike, and finally the harvest index. And as you can see, both high daytime and high nighttime temperature significantly decrease all of these components. And in general, for most of the traits, the combination of both day and high nighttime temperature was much more negatively impacted. And of course, I'm showing you 2 varieties here, the pink ones and the green. One of them is Carl 92, another one is Wettner. And then generally, Wettner is a relatively more heat tolerant. As you can see, the impacts on that variety was little less compared to that of a sensitive variety, Carl 92. So we do have genetic differences in response to both day and nighttime temperatures. Again, the processes which are impacted are the reactive oxygen species, which is represented here. In this visual, where the reactive oxygen species is much more produced in the sensitive variety, and the same thing is also with the MDH, which is also indirectly shows you the impact on the membrane stability. So those are the physiological processes which are impacted, finally resulting in lower photosynthesis and decreased seed set and smaller grain size. So question number 4 is that do high temperature stress have similar or different effects at various stages on different traits? And I've shown you that in the previous, while responding to the sensitive stages, that the most sensitive stages are flowering and a few days before flowering, which impacts the seed numbers, and then also the rapid grain filling stage, which impacts the seed size. So here's an experiment where those were very short periods of short episodes of heat stress, anywhere from 5 to 10 days of 5 to 2 days of heat stress. But now in these experiments, what we have done is we have exposed them to a little longer time in these different experiments. So in the first set of figures, you see the effects at anthesis, and then on the right, an extreme right, is the effect on grain filling stage. That means the entire duration there. So as you can see, again, at anthesis, the seed set percentage was decreased, leading to lowest grain numbers. And when the stress was exposed to the grain filling stage, the individual grain weight was decreased, and it resulted in lower grain yield. And then the smaller individual grain weight was due to two things. Number one is a decrease in the grain filling duration, because individual grain weight is a product of the grain filling rate multiplied by the grain filling duration. At a heat stress, the growing degree days accumulation is faster, and you generally see a shorter duration of grain fill. That will impact the grain size, and therefore that will impact the grain yield. And again, in terms of various physiological traits, you have the chlorophyll content, which is being decreased, and then the chlorophyll fluorescence, which is indicative of the photosynthetic capacity, that is decreased, and the thylakoid membrane damage is increased. All of these leading to decreased photosynthetic rates. And then the membrane damage was also due to the unsaturation index of these membranes, and I will come back and show a little bit more details on that. But again, it's all related to the membrane damage caused by reactive oxygen species and the unsaturation of those membranes. And you might ask that, what's the relationship between these various physiological traits and the yield components? So generally, we have seen a good relationship between the membrane damage and the photosynthetic. Higher the membrane damage, the lower the photosynthetic rates. And also there was a positive relationship between photosynthetic rate and the individual grain weight. More the photosynthesis, more the individual grain weight, because more carbon is available for increasing the seed size. And also there is a nice correlation between photosynthetic rate and the grain yield. But the one component which we did not see much relationship was the photosynthetic rate and the seed set percentage. So that shows that the cause for reduction of seed set is not the availability of the carbon, but it is more on the negative impacts of heat stress on the gamete viability, whether it is pollen grains or the ovule viability. So the question 5 is, what are the reasons for lower seed numbers and seed size under high temperature stress? And I've already alluded to you that the decreased grain number is primarily because of loss of pollen, loss of gamete viability. And I've shown you the data on the pollen, but also we also observe that the ovule viability is also impacted, and sometimes there is also early embryo abortion. All of those three components, loss of pollen viability, loss of ovule viability, and early embryo abortion, are the three reasons for lower seed numbers. And the seed size is primarily decreased due to decreased grain filling duration. So here's the data on the seed set from both primary spikelets, primary main spikes, and the primary spikes as well. And so in both of these, the high temperature stress decreased the seed set and the seed numbers. And on the right-hand side, I've shown you the effect of heat stress on pollen viability, especially now. Previously, I showed you the staining technique, but now here I'm showing you the in vitro pollen germination, because pollen germination is more important than the pollen viability itself. And again, there is genetic variability here among different genotypes. We have screened a lot more genotypes, but I'm showing you a screenshot or a sample of a few genotypes where we saw the impact of high temperature stress on the pollen viability, which was different among different genotypes. And then again, coming back to the yield components, here I'm showing you a response to nighttime temperature, where the nighttime temperature ranges from 14 degrees, 17, 20, and 23, and the spikelet fertility is decreased, which results in the fewer grain numbers. And then, but even in some cases, the total reproductive sites was decreased when the stress was too much. And then the individual grain weight decreased with increasing nighttime temperature. And as I mentioned to you before, the individual grain weight and the kernel weight was primarily a product of grain filling rate, which is the slope on this curve, and then the grain filling duration, which is the intercept where beyond which the grain weight does not increase, which I've shown you in different colors in these different four different component four different visuals here. On your left is the 14, top right is 17, and the bottom left is 20, and the bottom right is 23 degrees Celsius. As you can see, the rate was not that impacted. There may have been a slight increase in the rate, but that was not enough to compensate the loss of grain filling duration or the negative impact of grain filling duration. So it's a grain filling duration which is the primary driver of decreased seed size or the kernel weight, individual kernel weight in this case. So under high temperature stress, the primary impact is the decreased grain filling duration, and the slight increase in the rate sometimes you see is not enough to compensate the loss caused by shorter grain filling duration. And the second last question is there any genetic variability for key traits, especially in the wheat collections, wild relatives, and the chromosomal substitution lines? And we have screened all of these different available germplasm collections. So here is one of the screening which we did, and I'm showing you the data on the seed number, seed weight, and the harvest index. Again, as you can see, there is some genetic variability among the putative lines for heat tolerance for both stages, at the time of flowering and at the time of grain filling stages. And then the primary product, which is key when you're looking at stress tolerance, is the yield component, seed number, seed weight, and the seed yield, and the harvest index. I'm showing you harvest index, because it captures the thing which has happened during the entire crop production period. And then at anthesis, high temperature stress decreased the seed set and the seed numbers, and lower seed set was associated with decreased pollen germination And we were able to categorize these different germplasm collections into highly resilient, sensitive, and resilient types. So again, there is a genetic variability which needs to be exploited in the breeding programs. I know Dr. Ravi Singh will talk a little bit about what options do we have in terms of breeding for stress tolerance and also yield improvements. So that was among the putative germplasm lines. And then we also looked at different wild relatives here, the Aegilops, the Speltoides, Genicularis, McGraffy, and Cersei and Longishima. And then we found that there was significant difference in terms of the response of wild types to high temperature stress for the key field components, the grain numbers, grain yield, individual grain weight, and then and the grain size as well. And what we observed was that especially the Genicularis species was relatively more heat tolerant compared to others. And within those Geniculata, especially those genotypes which originated in Israel, Turkey, Morocco, were the ones which was relatively more are moderately tolerant to heat stress compared to that of other species. And of course, we were not able to make a direct correlation between the location versus the heat tolerance, but there were some species which were some lines within that species which were more tolerant. And then the same is also true for the chromosomal substitution translocation line. We had Dr. Vikram Gill has a whole collection of these, and we screened many of those translocation lines, and then found that there was differences in some of these translocation line in response to heat stress, particularly for the leaf chlorophyll content and the quantum yield of photosynthesis. Not only that, but also the individual grain weight and the grain yield. So we do have an opportunity for using many of these germplasm collections and use them in the breeding programs to improve the heat tolerance for wheat. And the final question is that how does wheat compare with other food grain crops in terms of cardinal temperatures and the responses? So this is a summary of my early career work. I had developed temperature threshold curves for majority of the grain crops, and I'm only showing you the seven crops here, but I also worked on few additional crops which I'm not showing you here. And I chose to show you the data on harvest index on your y-axis and the mean daily temperature on your x-axis. So this clearly shows the optimal temperatures for crops vary, and so does the ceiling temperature. Ceiling temperature is the temperature at which there is no formation of grain, so therefore the harvest index is 0. So as you can see, the wheat has the lowest ceiling temperature at 31, followed by rice at 35, followed by sorghum at 38, and pearl millet at 41 degrees Celsius among the cereals. And for the legumes, the dry bean is the most sensitive at 32, followed by peanut at 39 and soybean at 40. And a couple of other legumes which I have not shown you in this visual are cowpea and mung bean, which are relatively more tolerant even than soybeans. They have that ceiling temperature closer to 42 degrees Celsius, which is almost comparable to that of a pearl millet. So obviously, so there is a whole choice of crops which we can choose from depending upon your agroecological conditions. So you have to match the optimal temperature and the ceiling temperature based on your region so that we are able to choose the right crop for agroecology. So we do have options to choose from. So with that, I would like to conclude by most of these are general conclusion. Hopefully, I've shown you enough data that high temperature stress causes significant yield losses, including that of wheat, and even short episodes of high temperature stress, even 1 or 2 days of stress, if they occur at critical stages of crop development, will have negative impacts on yield through impact in the yield components, primarily the seed numbers and the seed size. And for most of the grain crops, including wheat, the most critical stages is few days before anthesis. That's when the gametogenesis and the viability of the gametes, both pollen and ovule, is being determined. And the second one is at the time of flowering, when the pollination, pollen tube growth, and fertilization is happening. And the third one is the rapid grain filling stage. That's when the grains are being filled. And the crops and the varieties within that crop species vary in their responses to stress and have different thresholds, and we need to exploit that. And there is a need to explore and systematically screen large germplasm collections, parental lines, and wild species for tolerance and use them in the breeding programs. And of course, there are several physiological, biochemical, molecular, and genetic traits that confer the stress tolerance. We need to make sure if you are looking at any biochemical, molecular, or genetic traits, we need to ensure that the functionality of the genes and the processes at the cellular, molecular, and biochemical level are relevant at the whole crop level and directly influence one of those yield components, grain numbers or seed size, and the partitioning through the photosynthesis. And of course, there are several traits, but the limitation is the high throughput phenotyping techniques. But now with the remote sensing tools with UAV, we may have some options for high throughput phenotyping and screening larger germplasm collection by using spectral imaging and also thermal imaging cameras put on these small unmanned vehicles to screen larger germplasm collections. But however, they need to be validated and integrated into the breeding programs as well. And there is a better need to understand the interactions of high temperature stress with other abiotic stresses, drought, salinity, and carbon dioxide, and few other climate change factors. Not only that, but also we need to look at how these high temperature abiotic stresses interact with the biotic stresses, the pests, the diseases, and the weeds. So we need to be focusing on interaction effects because at the end of the day, that's what is happening under the field conditions. There's multiple stresses which will impact. And the final point is that we not just only need to look at the impact on the yield, but also look more closely on the grain and nutritional quality of the nutritional quality, because as we know that not only food security is important, but nutritional security is important with a lot of micronutrient deficiencies happening, especially in women and children. So that concludes my talk. But before I leave, I would like to just put one slide on. I know I've talked about my work on wheat and how it is impacted by temperature stress. But over the last 7 years, I've been leading Sustainable Intensification Innovation Lab, which is funded by USAID, U.S. Agency for International Development. I know Dr. Ravi Singh is involved in another innovation lab, wheat genomics, applied wheat genomics, which is also at Kansas State University. So CEL, we call it CEL, is one of the largest innovation lab, and we will be active until 2024. And we have a large research education outreach programs in multiple countries in Asia, Africa, Latin America, and Caribbean. So the primary goal of our innovation lab is to provide access to safe, nutritious, and healthy food at all times to all people from existing farmland and without damaging our natural resources and ecosystem health. So the tagline here is the existing farmland and without damaging our natural resources. And to learn more about our work, please visit us on our website and follow us. So again, thank you so much, Dr. Khan and Dr. Ali, for giving me this opportunity. We are right at 30-minute mark. Again, appreciate the opportunity. If there's any questions, I can take it now, or if you have something else planned after the next talk, I can also do that at that point of time. Again, thank you so much.
Thank you very much, Professor Vara Parsad. So nice and presentation and very interesting and significant results, and we can see the hope, particularly once we see the heart waves in India and Pakistan. But you have shown us that how we can breed the heat tolerant wheat, particularly using the wild germplasm. So very interesting. The topic and the discussion and your presentation, well appreciated. So thank you very much. We will take a question at the end after the 3 presentations. So now I will invite Dr. Ravi Singh to please present.
Okay, are you going to share or should I try again? Maybe you share it to save time.
Okay, I think, SR, are you sharing?
Yes. Yes, let me try to share the PowerPoint you sent to me. And, let's see if it works. Then you can just keep calling next. So I will move to the next slide if it works.
Okay, sure.
I guess loading.
Still not coming.
Loading. Okay, so can you see the presentation on the screen, please?
Yeah, it came now. That's good. Can you put it on the full screen then?
Yes.
Okay, very nice. Yeah. So thank you very much, Dr. Khan and Dr. Ali for organizing the important webinar. And I like to share what we are doing to enhance yield, and of course that includes also yield under heat tolerance, under heat stress situations. So please, next slide. Can you change to the next, please? Yeah, to start, okay, go back, please. You got too fast. One back. Okay, so this, the next one.
I'm sorry, I think there is a lag between voice and video. So, okay, I will have to try to receive. So, is it correct slide?
Not this one, the one after this.
Oh,
okay. So what this figure shows is that over time, despite the climate change, you move too fast, Daksa.
I'm sorry, what I'm telling is there is a lag between, I think, your screen and the voice. That's why I couldn't properly follow. So this is the first slide of the presentation, correct?
Okay. No, so you have to go, yeah, here. So next time I will raise finger also. Okay, so you see me.
Okay.
Okay, so what I was saying that despite all the issues we discussed, we also need to see the positive aspect that global wheat production as well as yield shown in the red line there has been increasing over time. And if you think in the 60s, yields were around 1 ton per hectare, and now it has gone to 3.5 tons. So that is the good news. So next slide now. Can I have the next? And I wanted to also highlight the importance of Asia, in global wheat production. That means being the largest producer, if you think of globally, as well as consumer, whatever happens in Asia is going to have much bigger impact in other regions. So we have to ensure that we continue moving our yields, at least in Asia, and as much as possible in other parts as well. Next, please. Next. So I also plotted how Pakistan is doing, and as you will see on the top red line, the yield in Pakistan over the time has also been increasing, and the small table can show you the comparison between 1961 and 2021. So the area has doubled, obviously, but production and grain yield has increased several times. So that is very important part, and that is thanks to many different things, crop improvement, crop management practices, as well as policies. They all are important aspects to increase production. Next slide, please. So also we often look at how things have changed, let's say in different periods. So in this case, what I have done, I took South Asia here, and I have tried to compare the what was the average yield 10 years ago, taking 5 years average, and in the last 5 years. So what you will see that production has been increasing around 2.5%. 2.5% annually. Area has not increased that much, only 0.8% annually, though area is increasing. That means there is need for more wheat in that area. People are consuming more. But interestingly, what you see, the grain yield, the increase has been in the order of 1.7%, which is quite significant, if you think about. But I wouldn't say that's just because of the genetic yield gain, but once again, it's the genetic yield gain, improved crop management practices, and the policies. Next slide, please. Next. Next slide, please. So when we talk about wheat breeding objectives or wheat breeding, we cannot talk each trait on its own. And especially when we talk about genetic gain for productivity associated traits, we have to talk on yield potential gain, resilience to climate, which includes both heat and drought, and then durable resistance to major diseases and pests, because you don't do all of this together, somewhere you lose. On top of that, then in case of breeding, we also need to deliver genetic gain for market acceptability, and nutrition. And what I mean is end use or processing quality and biofortification. Also, we know, as Dr. Vara was showing, that even heat tolerance is so complex. And when we think of all these traits, what we also know that they all are complex and under quantitative genetic control. And that means we do require simultaneous selection strategy to develop better varieties. Next slide, please. So what we have to do is a very well targeted breeding to accelerate genetic gain. This is what we try to do at CIMMYT. So first there is define the target environments and product profiles for the selection of necessary traits. And then I think one of the most important part is the selecting high performing parents with genetic diversity for the needed traits if we want to make further progress. Then adopting appropriate crossing and selection schemes when you apply selection pressure, what methods you use. Then another key aspect is phenotyping for grain yield and other traits under well-managed selection environments and using appropriate trial designs. So phenotyping for making advancement is key here, so whether you do for yield potential or you want to do for heat tolerance or drought tolerance, we need to have good environments for selection. Also, as Dr. Prasad was talking about the role of different species, he talked about geniculotas, geniculata, etc. There is diversity within the wheat gene pool, the alien species, as well as genera. So we do need to maintain a good, what you could say, the germplasm enrichment program or pre-breeding type of activity where all the new traits or new sources of tolerance resistance are being continuously brought into the breeding gene pool. And then we need to explore new technologies as they come up. Something which is happening a lot in CGIAR nowadays for all crops is rapid generation advancement to shorten the breeding cycle length, as well as genomic selections. And Philomin will cover a lot on the genomic selections, and I will talk about the other part. So next slide, please. Next slide, please. So as you know, the CIMMYT breeding program has a much wider target areas in Asia, Africa, and Latin America. So we also have to do more targeted improvement to develop elite lines which could find adaptation in with our partners in those areas. So more recently, as I was mentioning in CGIAR, which is now called One CGIAR, for the last couple of years, there has been a major exercise to define our market segments and product profiles. So in case of wheat in the past, we were used to call this as mega environments, because we cannot breed for every small areas, but so we have basically grouped larger areas which have similarities into mega environments. And now that has been changed to what we call product profiles. So we have generated 6 market segments or product profiles which go for each of these 6 market segments. They are listed here. You could see in this table the area under each market segments. The largest is what we are calling now hard white optimum environment normal maturity. Most of Punjab of Pakistan and KPK area will fall into that. The yields are much higher on the average. This is considering the all environment. So this is just an information provided here because our yield trials and nurseries, which we will be distributing, would be based on these product profiles. Next slide, please. Next slide. Yeah. So for each product profile, we also have defined what are the key traits, as you could see in the left column of the slide. So that they could go from yield related, stress related, heat tolerance is mentioned, water use mentioned, quality, zinc, and lot on the diseases, as well as maturity type, because if you try to grow normal or late maturity types in let's say southern part of Pakistan, you will definitely suffer from heat stress. Like this year happened even in the areas where normal maturing wheats are sown. So we have developed this. We have also, you will see the size of the breeding program is mentioned, 2x or 1x size. So what is 1x size of program means that roughly 100 to 150 new crosses will be made for that particular each year for that particular breeding pipeline. So we have now 5 breeding pipelines in CIMMYT, which will be developing elite lines for each of the market segments. The last one here, the breeding pipeline 5 is going to take care of 2 market segments because the total area is not that big into that. And that's the only one where we have a red grain varieties or lines developed for certain areas. As you know, most majority of our target areas, they need white grain materials, and Pakistan is like that. Next slide, please. So this is the next slide, please. So basically, this is what is happening now in CGIAR for all crops. Everyone is doing this. Everyone is defining their product profile and market segment so they could target in a much better way the needs which people have. Okay, so many people know about CIMMYT shuttle breeding, which was implemented by Dr. Norman Borlaug in the 1940s. And what he did, he used diverse field sites in Mexico, you know, the Ciudad Obregón, the irrigated area where it usually doesn't rain during the crop season, and most years even throughout the year, the rainfall is very little. It's desert. So at that side, by controlling water, you could also create different levels of drought stress. By changing the planting time, we could generate different types of heat stress in the field. So that site is very important to us for phenotyping, for yield, drought tolerance, as well as yield under heat stress. So by using the Obregón and Toluca or Albatan, Toluca and Albatan are in the highlands, you had different disease pressures, but Dr. Bolog used 2 crop seasons per year. And the outcome of selection has been a wide adaptation in CIMMYT germplasm through photoinsensitivity, yield stability, multiple disease resistance, and more importantly, what was happening that it also reduced the breeding cycle time, and that means higher genetic gain. So this scheme we had a breeding cycle length of 5 years by using this shuttle. So keep that in mind that we were completing our breeding in 5 years. Next slide, please. So next one, please. So now the drive is to further reduce this breeding cycle length, and the thought there is if you could recycle your parents faster, then that should deliver higher genetic gain. So before I go there, this just shows that the Obregon field side, how crop looks. So we have there what we call managed selection environments. We have flat planting. We use drip irrigation, but you can get 9 and in good years, maybe 10 tons yield per hectare. We have a raised bed planting with flood irrigation. So we have 2 environments for to measure the yield potential, 2 environments for drought stress, and 2 environments for heat stress, early sown heat stress or juvenile stage, and then late sown heat stress. And you could do it side by side, and that is a big advantage over there. Next slide, please. Next slide, please. The change over time is slow. Next slide. Here it comes. So when we talk of genetic gain, we could start measuring right from the first year when the yield trials are done. And this is one example how the lines which we are testing in our first year yield trials has been shifting over time. So here, since 2013-14 season, we started to use a variety which is a very good example of variety which combines high yield with heat and drought tolerance, Bolog 100. You could see that in 13-14 season how the yield of lines were distributed compared to the last season. So you start to see that this bell-shaped curve for yields of the number of lines, which are also listed as N number, has been shifting towards higher and higher side over time. And when you change into roughly, it gives around 1.7% annual genetic gain. Next slide, please. Next slide. Okay. And we also monitor genetic gain in our international yield trials, which are distributed to all partners. So here is map of South Asia on the left. So South Asia, when you do the analysis of environments, it comes as in whole of South Asia, we have 4 major environments, which are these days also called for us for breeding target population of environments. So the biggest one is the irrigated optimum, which is has been pointed there, roughly 20.8 million hectare of spring wheat sown in South Asia. So if you measure the genetic gain in the yield trial called SWIT, Elite Spring Wheat Yield Trial, the analysis shows that the yield in the trial measured over that particular environment has been increasing at the rate of about 100 kg per hectare or 2%, which is quite significant. So the reason I'm showing this is important because breeding is delivering genetic gain. And I just put down there about Afghanistan, a similar trend was found in Afghanistan in this trial, same trial. They show a yield gain of 2.3% per year. And this is important. And this gives you thinking that we could continue increasing grain yield per hectare. Now the next slide, please. Next slide, please.
Yeah, Doctor,
Okay.
I'm saying immediately, but I think somehow it's taking some time to show on here.
Yeah, I know. It's just maybe internet is little slow. Sorry. So this part, what I'm showing you here, that going back to Breeder's equation, is happening in CGIAR, and hopefully it's going to be another lesson. It comes from the concept of speed breeding, let's say, that we are supposed to further reduce the breeding cycle length to increase genetic gain. So in Breeder's equation, obviously genetic gain is equal to selection intensity times selection accuracy, genetic variance. But more importantly, the breeding cycle length, which is the denominator. So it has been decided that all crop programs need to find ways how you reduce your breeding cycle length, and while trying to maintain your selection intensity and accuracy, as well as genetic variance. So next slide, please. So that is the part which is happening now in CIMMYT as well. And what we started to call a scheme rapid bulk generation advancement. So in this scheme, from that 5 years, which was used to take us, now we are trying to do reduced breeding cycle length to 3 years. But there's another interesting aspect here. We are not supposed to increase our cost. So in fact, they hope that our cost will reduce rather than increase. And if we reduce cost here, part of that cost could be moved to our national partners and support work there. So this is also another change happening. So it makes it quite complex if you think about. But what we have developed, considering we have 2 very good field sites, we are trying, the key changes here, if you see in the figure year 1, we want to make crosses, grow F1 and grow F2 in 1 year, first year. So, year 2, we go to the field where we want to do selection now. So first year, there is not much selection. You just make the cross and advance your F1 and F2. Year 2, we grow F3 in field in Taluka in this case, which are bulked, so your costs were cheap until then. And then select plants, make hedgerows, and grow F3 derived F4 hedgerows. So this is another change where people were used to think, okay, the last time you select should be F6 or F5 plants. We are cutting that by selecting plants in F3, grow F4 plots, and then in year 3, we have a season in Taluka Al-Batan, let's say again, to do whatever phenotyping we need to do and seed multiplication, which is required to do in the year 3 multi-environment yield testing. And then from there, recycle the best parents to the crosses. And year 4, from there onwards, goes towards product development. So we have built some field screenhouse facility in Taluka now, as well as some speed breeding facility to assist this. But I know the cost of a speed breeding facility is usually too high. So next slide, please. So to do the cost cutting measure, what we also are trying is, next slide, please. I hope it comes soon. Yeah. So we also like to see if we could do even cheaper in the field. So we have tried, this was the first season, which is still ongoing in the field. So we grew F1s by sowing early in our field site, Obregon. The upper figure, what you see, these are F1s growing there in the field. And you could see by around this year, we could plant on 24th October, and towards the January end, they were already harvested. Then we have below figure shows the F2s out of those F1s grown next to it. And we are expecting that they will be harvested by the end of this month, so we could grow the F3 in Taluka again. So if we successfully do this, and many other programs can also do it, you save a lot of cost because the speed breeding facilities are costly. Next slide, please. So the money you save in early segregating generation, you could invest in your phenotyping for yield. The other aspect, I will not go too much in detail because Philomin is going to do it, is the genomic selection in CIMMYT wheat breeding program. So Dr. Prasad mentioned about this innovation lab with Kansas State University. So we have program under this lab, which involves India, Pakistan, Nepal, and Bangladesh. So there has been a lot of learning on grain yield, stress tolerance, quality, grain zinc, iron resistance to diseases, and there are lots of publications. So how we are using it nowadays is to, based on the predicted values for grain yield and other traits, we could discard many lines without even testing them for the yield assay. So you use the predicted value and eliminate lines and save some money for yield testing. So when our advanced lines are being multiplied in Al-Batan, as I showed you in earlier breeding scheme, lots of them are eliminated and they are not going to yield trial. And Philomin will talk more about it. Next slide, please. So also the molecular data is very useful in developing the breeding values, which I hope Philomin will also explain. We also know that genotyping is very expensive. We were using the GBS genotyping by sequencing platform, which Kansas State University was providing us. But within CGIAR now, there's also a move towards the shared facility, which could reduce the cost of genotyping. So our colleagues are working with the DART company in Australia and selectively putting the SNP markers which are relevant for CGIAR germplasm, which is very similar to the germplasm you are using in many national programs. So it could be useful for everyone. And it's mainly for the genomic selection work at this time. But we are also trying to integrate all the known gene markers for disease resistance and QTLs, which people have identified for many traits. At this time, not everything is working well, but progress is being made. So this has been second year when people have been working on it. But one thing, what we have learned here, that coverage on different chromosome is not that bad. As usual, D genome has the least number of markers. But when you compare the genomic prediction using the old platform, the GBS marker, and this DART tag, you'll see for several traits in the bottom corner of the figures, what it shows is that there's very good correlation. That means this platform can be used in the future and cost will be much cheaper, perhaps $13 per line, including DNA extraction. Next slide, please. Next slide, please. And we have to do this because I think if we could integrate all the known gene markers on it, then you get very good information on the QTLs, important QTLs and disease resistance genes for which markers are available. Yeah, so talking on the disease resistance, our main focus, main objective here is on durable disease resistance to achieve resistance durability, avoiding epidemics, and minimizing chemical control. Could say the rusts are still the main diseases, but depending on environments, we also work with septoria, spot blotch, tan spot, fusarium, wheat blast, which is in South Asia now, as you know, in Bangladesh. And there are other areas which are prone in South Asia if disease spreads, as well as we do some work on kernel bund. Next slide, please. So while slide is changing, one of the reasons why we work on resistance durability instead of using race-specific major genes or try not to use them as much as possible is because pathogen population is evolving very fast. And to achieve a sustainable disease control, we also need to have research areas to learn pathogen diversity and changing epidemiology in climate change. We have to have good resistance phenotyping, whether you do under a greenhouse or in the field condition, genetics of resistance, mapping of new genes and have molecular markers, gene cloning, very important for future, especially for of course, to get the best markers, but also now for gene editing purposes will be very important if more and more genes are cloned, and then breeding for quantitative resistance. Next slide, please. Next slide, please. Yeah. So my experience, why we started to focus on race on the durable resistance goes back to the boom and bust cycle. And there's some example with leaf rust that you know, the year varieties were released, and table shows that few years, 3 years, 2 years later, 4 years later, a new race was identified. And that was the time when variety was picking up most of the area in northwestern Mexico. But that means pathogen population, in this case, leaf rust, was very dynamic and continuously evolving. And the last case, the variety Baviacora, the resistance there broke down because of the introduction of migration of a new race. And this race in North America has become very important and has many other biotypes. But one of the success stories has been that after 94 breakdown, we didn't have any more resistance breakdowns, and that's because we shifted our strategy to adult plant resistance based on minor genes. Next slide, please. I think in the next slide, I have just very quick example, I hope it comes soon, of how the UG99 race of stem rust was identified and started to migrate very fast and evolve at the same time. So next slide, please. So when we started with that in 98, 99, there was only one race which was identified as UG99 and was called UG99. Now we have at least 13, and very recently I hear there are a couple of more variants they have recently identified. Next slide, please. It's taking a lot of time, sorry. But anyway, so UG99 race has.
Hello, Dr. Ravi, can you listen? I think there was an interruption side. Am I on the correct slide?
No, one after this, please. In fact, you can move two because time is running out very fast.
Okay.
The next one was the slide shows how UG99 has migrated across Africa. And the one after that, I believe, shows the how in climate change since 2000 onwards, the yellow rust, which has become problem again in Pakistan, the variety Faisalabad 2008 and several other popular varieties became susceptible. Yeah, stay on this one. So we skip couple of slides, but that's fine. So the strategy for us nowadays for rust resistance is to use genes which individually don't give large effect, but their combinations give you a high level of resistance. We call these genes are called slow rusting genes, and some of them which are listed here like LR34, 46, 67, even SR2 gene for stem rust, they give resistance to more than one disease, but partial resistance. So strategy here is combine 4 to 5 of these QTLs, if you want to call them, and that gives you much higher level of resistance. And very similar mechanism operates for many other diseases. Next slide, please. So this has become the basis of adult plant resistance in our breeding materials. So if you could move to the next slide, please. Yeah. And we also, another thing which is being done now is the part of the germplasm enrichment is to use the speed backcrossing. into these speed breeding rooms, and some of the genes which we are very quickly introducing in combinations and not alone. The idea is to have at least combination of these race-specific genes, which tends to be more effective using molecular markers. But also you run out of the genes for which you have good markers. So there is much more work needs to be done to develop good markers. If marker-assisted backcrossing needs to be successful in the long run. Next slide, please. I will have just a couple of slides on biofortified wheat varieties. You know, Pakistan was first one to release from the Harvest Plus project variety Zincol in 2016. Zincol was good variety, still grown, but perhaps was little bit behind in yield. But now the variety Akbar 2019 combines the high yield as well as high zinc. And I understand it's spreading very fast in Pakistan, which is a good news because then people are benefited with high yield as well as high zinc. And a new one was recently released last year, Nawabi 2021. Interesting part, you will see in zinc, all the source of high zinc was Triticum spelta. In Akbar, it was a variation within the elite breeding parents gave the high zinc. And in Nawabi, the high zinc is coming from a Mexican land race. So there's here component of pre-breeding, if you want to call. Next slide, please. And now our program is determined to make zinc as a mainstream trait. Means in next 8 to 10 years time, all breeding lines will carry high zinc to benefit the zinc deficient population, which is very high in Asia and Africa. Over 2 billion women and children are zinc and iron deficient. So we have we have all the fields in Obregon. I mean, soil has been homogenized and we have started to measure zinc in lots of materials now, which is aided by this XRF screening method, the machine which you see. You could do 250 samples per day through non-destructive technique. And the graph which is shown on the right side, there is a lot of variation within the breeding materials we have. And the push which we are giving now is to choose parents which combine high zinc with high yield. And that means you shift the zinc and iron values, and zinc and iron values are correlated, by the way. So you select one and you get the other. Next slide, please. I think many of you know how we every year distribute our every elite lines which we have developed. They are distributed through different trials and nurseries, as well as for diseases. There are nurseries being distributed between 50 to 170 sets to more than 65 countries. Germplasm goes, people have to request them. Every year, a request has to be filed in through online system and materials are provided. Next slide, please. And while you are changing, this is very important part, you know, that people are getting and growing and providing the data, which is very useful to everyone. In this slide, something happened, half got kind of covered. Good now. So while we are trying all from CGIAR side and your side to release new varieties, I think one very important area is how do you accelerate the seed dissemination of new varieties? You know, in the last some 6 years, from 2015 to 21, 7 years roughly, 48 varieties were released in Pakistan. 38 were selected from one of the international trials and nurseries out of that, and others were developed locally. But I also know that only a few varieties become popular. As you know, only a few cover millions of hectares, and some of them never reach farmers. So we need to put, and this is area I really hope every country, including Pakistan, puts major effort to ensure that released varieties reach farmers faster through pre-release seed multiplication, accelerated and large-scale post-release seed multiplication. Low density precision sowing can be used for this. So you could use 15 to 20 kg seed rate per hectare and you will get roughly the same yield. Engaging private companies and farmers group in seed business and dissemination. Seed should be looked like business, you know, and then only you will see. And I think with Akbar 19, the way it's spreading, we should learn what has happened, why Akbar 19 is moving fast, and maybe a similar model can be applied with some further adjustment. And you know why? Because just, you know, sometimes we think, oh, 1% yield gain, what, it's not much. But when you put a value on it, let's 27 million hectares are sown in Pakistan to wheat. So if you change those 27 million hectares, you had 270,000 tons of grain produce worth 108 million US dollars, considering current prices. So we should not be losing this value, you know. And that's why it's important that new varieties, hard work from the breeding gets into farmers' field as soon as possible. And we maintain varietal diversity in farmers' field. That's other aspect. Next slide, please. I hope the next one is the last one. Yeah. So to conclude, I'm very optimistic because yield potential has continued to increase. However, further reduction in breeding cycle time combined with approaches to improve parental selection could give us even slightly higher genetic gains. And we do need to package yield potential with tolerance to and resistance to multiple abiotic stress. I think in climate change, you cannot breed one trait, you know, all needs to go together. And of course, resistance durability we could achieve by releasing and growing new varieties that possess complex adult plant resistance. Next slide, please. And I think that's just the acknowledgement to different donors. And we also have training programs to with many different partners. Unfortunately, the in-service training program for the last few years has been suspended to online courses, but hopefully from next year we could restart our proper training. And thank you very much. And sorry about taking a little longer. It took longer to change the slides. Thanks.
Thank you very much, Dr. Ravi Singh. Very interesting presentation. Though the technology was a hurdle to properly listen, but it was really very interesting, and there is you can say a lesson, and that how we learn from the experience historically and how we can go ahead. So thank you very much. This time, Professor Parsad took benefit smartly and answered all the questions which were people they were asking in the chat box. So thank you, Dr. Ravi Singh, for this interesting presentation. Now I request Dr. Philomin Juliana. She is a wheat breeder and a quantitative geneticist in the Borlaug Institute for South Asia. So over to you, Philomin Juliana.
Yeah, thank you so much, Dr. Ali. Is my slide visible and am I audible?
Yeah.
Yes.
Yeah,
Yes.
okay.
Yes.
Yeah, thank you so much for giving me this opportunity to present today. So I'll be today talking about the application of genomics and quantitative genetics in wheat breeding with the CIMMYT Global Wheat Program. And I work with Dr. Ravi Singh and Dr. Arun Joshi in BISA. And this is a collaborative effort done by a lot of CIMMYT, BISA, and Kansas State University colleagues. Okay. So this is the outline of what I'll be talking about today. So my major focus today will be on genomic selection. So this is used by the CIMMYT Wheat program to increase the selection accuracy and to reduce the cycle time, as Dr. Ravi was explaining. And then utilization of markers that are identified in association mapping studies and also in QTL mapping studies to increase the selection accuracy. And then the third thing I'll talk about is about allelic fingerprinting. So to select parents, now we have a lot of information on parents for the different alleles they carry, let's say for grain yield, for disease resistance. So how do we make informed and accurate selection of lines and parents using that data? And the next thing that I'll be talking about is the utilization of selection indices. So as we know, to select breeding always involves multiple traits, and to select for multiple traits, it might be difficult, cumbersome if we have a large number of traits that we are looking at, like in the CIMMYT breeding program. So selection indices have been used these days to get the breeding value of individuals and also to increase the selection accuracy. And then I'll also briefly touch upon what we do to how we apply genomics to find the progress in breeding, to understand the effect of selection, how the favorable allele frequencies have shifted over generations, and then about using some quantitative genetic parameters like marker-based heritabilities, genetic correlations, and response to selection, direct and indirect selection. So all these parameters are helping us to understand what's going on in our breeding program and also to optimize it and make efficient selections. So I'll briefly talk about what genomic selection, what is a strategy used for genomic selection. So it contrasts to the conventional marker-assisted selection because now we have markers distributed across the whole genome. So the first step in genomic selection is training the prediction model and evaluating the prediction models. So this is just any set of existing breeding germplasm which has diversity for a trait of interest. So here, for example, I'm taking irrigated grain yield in Mexico, and if it should just has variability for the trait of interest, and we take about 300, 400 lines are more than enough for testing genomic prediction. And this can be any existing set of lines. So that is lines which have both phenotypes and genotypes. So that is called as a training population. And this data is used to train prediction models. So we use both phenotypes and genotypes, and then we predict the performance of another set of individuals known as a validation population. And then we make a correlation between the actual grain yield of those individuals and the predicted grain yield. So that is known as a prediction accuracy, which I'll be talking about a lot in the coming slides. So we estimate the prediction accuracy. And then in a real genomic selection program, you would just train prediction models, obtain the genomic estimated breeding values of new lines. So instead of putting the lines in the field, we would be predicting the performance of the lines and then select the lines which have the highest genomic estimated breeding values, make crosses, and advance generations. So this is the procedure of how we implement genomic selection. And this population is just called as a breeding population or selection candidates. And one key criteria is here is that the germplasm in the selection candidates should be highly related to the training population. Only then we would have good predictions. And then the conventional process of line testing and variety release is there. And in the CIMMYT wheat breeding program, I'm going to show you at what stages of the breeding program we apply genomic selection. So in the selection of parents, the genomic estimated breeding values of individuals can be used to select parents for crossing. And then we have a large number of lines in the early generations where the selected bulk is used to advance breeding scheme is used, and it's used for advancing generations. So each generation you select the best individuals and you bulk about them. So here there are large number of lines, about 0.4 million plants in the F1s and more than about 2 million plants in the F2s. So at all these stages, although genomic selection is very good for differentiating all these sister lines in early generations, but still the population sizes are too large for genotyping and there are logical constraints. So we do not implement genomic selection in this stage. And also there's high heterozygosity and there is segregation in the early generation. So again, we do not use genomic selection for these early generations. And one other reason why we do not apply here is that there is a low cost benefit ratio. So unless genotyping, the current genotyping cost could vary between $10, $15, $20 for genotyping by sequencing. And unless that cost comes down to the cost of taken to advance from crossing to the hetero stage, again, we might not be able to have a very good benefit of genomic selection. So cheaper genotyping platforms are needed to apply genomic selection in large scale breeding programs where there are lots of early generation progenies. And then the next stage, we have, again, heteros still greater than 70,000. So here also we do not implement genomic selection because of the high cost of genotyping. But then we use a combination of pedigree and high throughput phenotyping selection in this generation, which we have been testing. And then in these two generations, so as part of the USARD project, a large number of lines, about 9,000 lines each year, have been genotyped for more than the past 8 years from 2013 onwards. So here we have phenotypic data on grain yield, agronomic traits, and stem rust. So we evaluate genomic selection for these traits here. And this is a stage where we extensively have evaluated genomic selection. So these are about 1,000 lines, which are selections from the first year of yield testing. So these are called the second year, stage 2 of yield testing or elite yield trials. And then we have lots of data for grain yield in different environments, diseases, and quality. So these are the two stages where we have evaluated genomics selection in CIMMYT program extensively. And so as I was just telling you, so this figure just shows more than 70,000 lines for the past several years have been genotyped as part of the USARD project and the delivering genetic gain in wheat projects. And these were all genotyped using genotyping by sequencing markers. And Jesse Poland's lab from Kansas State University was involved in this genotyping. And just moving on directly to the results, what we have for genomic selection. So we have made two kinds of prediction scenarios. So one question we are interested is within panels, within breeding populations, and within a single year, how good are we in predicting different traits? So this figure shows just color coding the traits by quality, which are all in red here, and then by diseases, which are all in blue, and then phenology, heading, and maturity in orange. And then we have grain yield in late sown, early sown, drought stress, and irrigated conditions in green here. So the accuracies here, these are the genomic prediction accuracies for the different traits, the mean of different 4 different seasons of evaluation. And in each year, we had about 1,092 lines. And about 800 lines were used to predict the remaining 200 lines. So this was done using multiple repetitions of sampling. And then you can see the best predicted trait was grain color, very obvious because of the simple nature of the trait. And then a lot of quality traits, again, were very well predicted. For example, mixing time, 0.73 was a prediction accuracy. Alveograph W value, again, 0.7. And then stem rust in Kenya. This is adult plant resistance in Kenya, which Dr. Ravi was talking about. So this is, again, predicted very well. And then we also have grain yield and heading maturity, which was predicted about 0.5. The prediction accuracies were close to 0.5. And then now we also test another scenario called forward genomic prediction across nurseries and environments using historic training sets. So let's say for this cycle, we have 1,000 lines in the field and we want to predict which are not in the field, and we want to predict the performance of these 1,000 lines even before they are in the field. So we can use a lot of historic data. So here in this case, we had about 3,000 lines which had historic data. So we used all the 3,000 lines to predict a new set of 1,000 lines, and these are the prediction accuracies. Again, you can see stem rust well predicted, and a lot of quality traits, again, well predicted. Alveograph still is high, mixing time still high. And then you can see grain yield. You see this big dip. So I'm moving on to the next slide where you can compare predictions for within panels or within environments and across panels or across environments or years. And you can see clearly for grain yield, if you predict within years, the prediction accuracy is about 0.5. And then if you start to predict across years, it dips down to 0.25. 0.3. So that is a very good indication because it's a very highly hard trait which is highly influenced by genotype by environment interactions, and prediction across years is quite challenging for grain yield. But still, stem rust and a lot of quality traits were well predicted. So that was our take home here. So end use quality and disease resistance rate are well predicted from historic training sets at CIMMYT, but then grain yield predictions for unknown future environments is challenging. And this is another slide which just shows how we do genomic selection and compare that to phenotypic selection. So if we just select the top 10% of the lines from our stage 1 yield testing, which has about 8,700 lines, and we select the top 10, 870 lines, and then if you just make predictions within the year, the accuracy was 0.52. And if you use prediction from the previous year for all these 8,000 lines, the accuracy was 0.3. And then we made a cutoff. So if you just select the top 10%, so you can see the green portion here shows the lines which are selected by genomic selection and phenotypic selection. So about 28% of the lines, if you use the previous year data, can be selected by both these methods. And if you just use within the year data, then about 36% of the lines can be selected by both these methods. And if you just consider this part, the huge red part here, these are lines that are discarded by both genomic and phenotypic selection. And you can see a large number of lines, about 93% of the lines are discarded by both these methods. So this is a very nice indication that if you just use genomic selection, you might end up losing a lot of top performers. So for example, you can see based on the observed grain yield, all these lines were above the cutoff, the top 10%, but a lot of lines in blue here were just selected by phenotypic selection, but the GEBVs or the genomic estimated breeding values, the predicted grain yield was very low. So if we just rely on genomic selection, then we might be at the risk of losing about 65% of the lines as well. So we always take that into consideration, and considering that risk, genomic selection is always integrated with phenotypic selection whenever possible. And then this is another thing that we are doing as part of the USAID project. There's a new trial that was developed several years ago called the South Asia Bred Wheat Genomic Prediction Yield Trial. So this is showing data predictions of these trials in different target population of environments of CIMMYT. For example, Faisalabad, Pakistan, Ludhiana, India, and Nowshera, Pakistan, they all come under the target population environment 1. And then Bhairava, Nepal, Jabalpur, Pusa, Sindh, all of them come under the target population of environment 2. And then Jabalpur in India comes under the TPE3. And you can see grain yield prediction accuracies are quite variable. And this is just for the last cycle. And if you see the previous cycle, it might be different. So there are a lot of environmental factors, management conditions, which affect the prediction accuracies. But some years you can see, for example, last year, Faisalabad had a prediction accuracy of 0.26, but and similar was Nowshera. And Sindh pretty much similar. And some sites like Jamalpur, the prediction accuracy was high. So we have to expect different prediction accuracies in different environments, depending on the heritability in those sites, management conditions, etc. And to understand this, we also use quantitative genetic parameters. So what is happening? Why do we get low prediction accuracy? So then we also try to find out the trait heritabilities in these different sites. So for this, we use marker-based heritability estimation. So we use genomic data. So you can see in different locations, let's focus on Faisalabad, Pakistan. You can see the genomic prediction accuracy was 0.26 and the heritability was only 0.3. So when you have low heritability across the replications, then that's exactly what you would expect in the prediction accuracy as well. So only the heritable portion of that genetic variation can be predicted. So that is a very nice indication that we also understand using heritability estimations. And then early eval. So this slide is showing us a large number of lines are evaluated, CIMMYT candidate wheat variety lines are evaluated in the target population of environments. So in this cycle, you can see locations Faisalabad, Thandojam, and Nowshera, and this is all coordinated by Dr. T.P. Tiwari from CIMMYT Pakistan. And you can see a lot of these South Asia bred wheat genomic prediction yield trials targeted for target populations 1 to 3 are evaluated in Faisalabad, Nowshera, and different sites in Pakistan evaluate different of these trials based on which target population of environment they're coming under. So each year, new candidate CIMMYT wheat varieties are evaluated in Pakistan and several South Asia countries as part of this project. And this year, we have also expanded the phenotyping network and also genomic selection in these target population of environments. So we are awaiting data coming from all these different sites. So for last year, we just had Ludhiana, Jabalpur, Pusa, and we had Thandojam, Faisalabad, and Nowshera from Pakistan. But this year, we also expanded to Chakwal, Bari Chakwal in Pakistan. So all these about 20 sites and CIMMYT's target population of environments in South Asia, we get the data. And genomic prediction will be done in all of them, and the GEBVs will be delivered to the national partners. And this information is also used by CIMMYT breeders. For example, you can see here, we 1 year before the lines are evaluated in these target environments, we estimate the genomic estimated breeding values, and it saves 1 year of time before the actual phenotyping. And then you can see here, we use different training sets. We use cycle 1920, cycle 2021, and then whatever available information we have for the different target sites. Let's say Pakistan evaluates from cycle 1920, we have the genomic estimated breeding values. From 2021, we have the breeding values. And then from all different sites, we estimate all these breeding values and deliver them to the breeders for selection a year earlier so that they can target lines to the target population of environments. And then we also developed a selection index. So this is just showing an index for TPE1, which has sites like Faisalabad, Ludhiana, and Nowshera. So now you can see we used an index called the eSIM, again, selection index method. And then you can see lines that were selected by this index were very much correlating with Ludhiana and Faisalabad, but that Nowshera that year had a quite lower heritability, so the index also takes that into account. And you can see very nicely a combination of grain yield from different sites, which are fed back to the breeding decisions. And then one of the success stories that we have for genomic selection in CIMMYT is that we've used it for scaling up quality selection. So that's what I've showed you before. All these traits, alveograph traits, a flour protein or flour yield, have very high prediction accuracies, at least very reasonable prediction accuracies. So we use the historic training population, and then we predict all these lines based on them. So the cost of phenotyping for quality is going to be about $61 per sample. And then the cost of genotyping is again close to $10 to $20 per sample. So if you are just predicting all these lines, so just subtract the genotyping cost from the cost of phenotyping. So you're still saving about $414,000 each year if you just go for genomic selection. And it's also practically impossible to probably have 8,000 lines and have a quality lab that can analyze all these different lines. So there are very unique conditions where genomic selection can help us. But for traits like grain yield, we still have to use phenotypic selection in combination with genomic selection. And then we also use genome-wide association mapping approaches. So we've tried to see, is there a shared genetic basis between yield that is evaluated in Obregon, Mexico, and in the target environment? So then we were doing this GWAS study with grain yield from Mexico, Ludhiana, and then you can also see Bahawalpur, Pakistan, Islamabad, Pakistan. So we had a lot of SWIT data. So you can see the 2NS translocation was clearly associated with yield in all these different locations. So we look for common genomic regions. And 2 cycles before, there was also striped rust evaluated at CCRI Pakistan and Faisalabad Pakistan. So that information is also, we also try to find out what genomic regions are associated and if we have more evaluations and how do they change across years. And all that information helps, genomic information helps us to monitor all those different changes. And Dr. Ravi also mentioned this. So we've been doing meta-GWAS for large number of traits in the CIMMYT wheat breeding program. So all the traits, let's say be it blast or leaf rust or spot blotch, stripe rust, or all the quality traits or grain yield, they've been able to associate, find significant markers associated with all these key traits. And all this information, genomic information, is helping us to strengthen the current marker-assisted selection strategies. And we've also developed a reference map. So this is the international wheat genome sequencing, the reference genome of red wheat, the RefSeq version 1. So we put all the maps that markers that were significantly associated in the CIMMYT wheat germplasm along, let's say, with the major genes. So we know this marker, and we know that there is a major gene SB3, spot blotch 3 gene, or SR2 gene. So we know all these relative positions now for most of the traits that are evaluated in the CIMMYT wheat breeding program. And we've also done fingerprinting of our lines. So as I was mentioning before, so let's say this particular marker was associated with grain yield in Islamabad. And then we have that marker here. And then about 44,000 wheat lines. So each one of these columns represents 44,000 wheat lines. And the presence of a favorable grain yield increasing allele is in green, and the absence of that allele is in, or the decreasing allele is in red here. So you can see if for a particular marker, let's say this 3B marker, which is associated with grain yield in Islamabad, we know what all the CIMMYT 44,000 CIMMYT lines carry, which allele the CIMMYT lines carry for this, for this particular marker. And it's the same here. So this again, let's say for A, Bahawalpur, Pakistan, we know a lot of CIMMYT lines are not carrying the favorable allele. So this kind of information, we've already developed these fingerprints, and we're able to look at it location specific, or some markers are associated with multiple locations. But you can see this marker, Ludhiana, Pusa, and Bangladesh. So those are the markers that we are interested mainly, because we cannot be selecting at each marker. And that's why we go for genomic selection as well. And then we also try to understand what is the effect of selection. So these are all 47 grain yield associated markers. And then you can see the elite spring wheat yield trial. So you can see all these yield trials from 2003 to 2017. You can see the alleles are they fixed? Are they close to fixed? So you can see this 3B marker, a favorable allele for grain yield is close to fixation. And this 4D has drastically increased. The 2NS translocation here has drastically increased. So all these trends we also analyzed using genomics data. And Dr. Ravi was mentioning about this. So we have the international wheat nurseries distributed, and there are a lot of requests. So this year, to 2021 to 2022 cycle, there were a lot of requests, more than 170 for CIMMYT wheat nurseries from Pakistan. And you can see a lot of these, a lot of this is coming from WRI Faisalabad, and then Islamabad, and then Bahawalpur, Pirsabag. So a lot of requests for CIMMYT nurseries, most of them from the government sector and some from the educational sector, and very little bit from the private sector. And this figure shows the grain yield in the SWITs from 2003 to 2017. So this is an example of how the data returned back from partners, national partners, and Pakistan is helping the CIMMYT breeding program. So you can see a lot of these locations have evaluated the SWITs over the years. And again, you can see some years is some locations, especially Islamabad, you can see the, again, combination of genetic and agronomic practices. You can see yield increases. Some locations yields have been quite stable, but then there are all trends of yields, again, which highly insist that developing climate resilient wheat varieties is critical. But there's also a good positive thing. For example, you can see the SWIT lines with the highest yield in comparison to the local check. You can see that line, the SWIT lines across the years from 24th to the 37th SWIT. You can see in Islamabad, Pirsabag, you can see always the highest leading SWIT check was above the local check. So there's always a very good impact of CIMMYT varieties, and it's bringing an impact in that region. So we also, again, estimated heritabilities for all the sites in Pakistan, which evaluate the SWIT. So we're using markers to estimate this. And you can see the heritabilities going on from 0 to 1. And in some sites, let's say Faisalabad, for example, you can see the some heritabilities for the SWIT nurseries could be 0.3 earlier, but now they have highly increased to 0.88, 0.85. Again, showing very good evaluation of trials in different Pakistan locations. And we also do genetic correlation estimation. So you can see from the data that we have, let's say we correlate all the other sites in South Asia. For example, this was from the last year. So we see which other site in South Asia has a high genetic correlation for grain yield with Faisalabad. Let's say last year, Jamalpur in Bangladesh had a 0.6 genetic correlation with the yield in Faisalabad. So this again changes year by year, but this is a very interesting trend to see how locations cluster every year and how do they change. Again, Sindh was a unique location. Faisalabad was quite separate, but then Jabalpur was highly coinciding with CCRI KPK. And then we also see the genetic correlations between the sites in the national, between sites in South Asia and also Obregon. So we have bed 5 irrigation, early heat, drip, late heat. And then we see, let's take Faisalabad again, for example, we see in this particular cycle, Faisalabad had a high correlation with 2 irrigation environment, selection environment in Obregon. So that's also helping us to understand what's going on between the selection environment in Obregon and evaluation in the target sites. So again, I'll just go to the conclusion slide because for the sake of time. So I'm just in summary of what we've done, what we've discussed so far. So in all the early generations that I've mentioned, CIMMYT, we do selection selected bulk scheme. And then we have the small plots, the pre-yielding stage. And last year, we had about 18,000 lines from this stage, only genomic data, and we completely used genomic selection. And we also used a combination of selection indices. And then the stage 1 yield trials, again, 8,000 or 9,000 lines with genomic and phenotypic data. We used genomic selection for traits. Let's say, for example, wheat blast, where we cannot phenotype 8,000 lines. There we use genomic selection. Or wheat quality, where again, we cannot phenotype a large number of lines. We use genomic selection. But for yield and other traits where we have the phenotype, we just use a combination of phenotypic and genomic selection and then use them in selection indices. Always we apply GWAS and quantitative genetics. And it's the same with all the other stages of yield testing as well. And with that, so the outcome of the stage, all these stages of yield testing and all this genomics and quantitative genetics works is again, what comes as an international SWIS with nurseries, with a distributor, the partners. And with that, I would like to thank you very much for all your interest. And I would like to thank all our major partners, the Gates Foundation, UKAID, USAID, FFAR, Kansas State University and Cornell University, and most importantly, all the national partners who generated most of the data that I presented today. Thank you very much for your time. Thank you.
Thank you very much, Dr. Juliana. Very interesting presentation, specifically handling large populations and large data points. So thank you very much. We move to question answer session. Most of the questions are related to high temperature or heat has been answered by Professor Parsad. So thank you very much, Professor Parsad. You have already answered most of the questions. So a few questions they are unanswered. I will just read from the chat. One is for the Dr. Ravi Singh. Dr. Ravi, speed breeding is one of the advanced technologies for speed growing of many generations within short time. But it has been reported by some scientists that controlled light along with other growth promoting conditions do not give complete opportunity for genetic potential to expose the traits which could be of advantage, and such genotypes may get unforeseen during selection of elite plants from pulse. So
yeah, okay, thanks. I saw that question and I thought, should I respond there? That is better. So if you see the way we have described our breeding scheme is a bit different than what often many think speed breeding needs to look like. So I think maybe unnecessarily people say you need to grow 6 generations per year or things like that. What we have to do is to fit in a component of speed breeding into your own breeding scheme to accelerate your own whatever breeding objectives you have. In our case, because we have 2 field cycle, we need to advance fast, just 2 generations, you know. So if I can do, we drew many schemes, but we cannot do in less than 3 years anyway, you know, because wheat is self-pollinated crop. It could be different in some hybrids. So that misconception needs to change. Where you which generation or population you pass through speed advancement, which one you don't. Then going back to, if you even follow the speed breeding, it's a bit like single seed descent. Just you are doing single seed descent in a much faster manner. So if you sow, let's say 200 plants per cross, you harvest a grain from there, that is spike or 2 grain, and you advance like you do in single seed descent. So it should not affect the structure of population because your sampling will come from those 200 plants. Because I also know some plants you might miss every time you advance, you lose certain percent. So that's my response. I have not done myself that level of speed breeding because we don't think that is good for our purpose, you know. So and I think you think about it, what is speed breeding? Speed breeding is not just advancing a generation faster, but how you have good phenotypic data generated in shorter time, and you, with confidence, you could recycle your parents back, you know, and that's what CIMMYT has proposed, and this is what is happening in CGI-AR. Thanks. Maybe Dr. Vara Prasad, you may, from university side, you may have heard people saying 6 generations, 5 generations per year. That's
in addition to the speed reading, one of the other advantage for wheat is the availability and use of double haploids. So I think that has another opportunity to increase the generations rapidly.
Yeah, I just like to add on double haploid. In case of wheat, for the cost of double haploid hasn't gone down, and that restricts, and that's why our donors and CGIAR has gone on a cheap population advancement approach as much as possible.
That's right.
Yeah.
I agree. That's correct. Yeah.
Thank you, Dr. Ravi. Another question was the iron biofortified varieties, I think in Pakistan or somewhere else.
Yeah, so in case of wheat, what happens, I guess that question is to me. We say more on zinc because zinc is more bioavailable when you consume than the iron. So even if we increase the iron in grain, most of it is just not enough. So that's why it's not talked about. In this genetically modified rice is coming up in Philippines where iron and zinc will be very high, and maybe then only you get the benefit. What we have learned, if you increase the zinc, iron increases by itself. And when you do the mapping work, the Jivas and other, there are a number of genomic regions are same. So they work in favor of mobilization of both zinc and iron. But there are other regions which work only for one or the other. So even if we increase iron, it's not going to benefit much at this stage.
Yeah, thank you, Dr. Ravi Singh. I think one question is Parsad. In Pakistan, heat wave mainly comes at antithesis stage to onwards. I want to know which key traits, 2, 3, we should focus to breed heat tolerant wheat genotypes.
Thank you. Thank you very much for that question. I just responded in the chat box. So if you are getting heat stress during antithesis, and as I mentioned at antithesis or a few days before antithesis, one of the most significant impact is on the viability of the gametes, whether it is pollen or ovule, which results in lower seed set and lower seed numbers. So the traits which you must be looking at is potentially getting higher seed set, and those which have higher pollen germination or pollen viability under heat stress. That's one option. And the second one is, I don't know, Dr. Singh can probably let me know about this, because there may be a genetic variability and time of day of flowering. I think we did find some genotypes of wheat which flower very early in the morning. So what they're doing is they're actually escaping the midday or 2 p.m. heat stress. So once the embryo is formed, it stays there and it's relatively more tolerant. So this trait of early morning flowering has been experienced and done well in rice. So there are certain genotypes of wheat which flower early. So that is another escape mechanism which could be utilized. And the third one is a stay green trait or the longer green leaf area duration, which is always helpful, and also higher biomass at flowering so that it has more opportunities for partitioning the carbohydrates into the grain when you have stress at the time of flowering. So these three would be the key items. And we have also looked at respiration, because if you're able to optimize the respiration, you'll have opportunities to increase the yield. For many years, we have just focused on one aspect of the partitioning, that is photosynthesis, but we also need to look at opportunities for respiration. Of course, the measuring respiration is difficult, And that's the reason why we haven't done much work, but there exists an opportunity to ensure that we are optimizing the net carbon, which is the difference between the gross photosynthesis and the respiration, which is available for growth and yield. Thank you.
Yeah, thank you very much. I think no more questions. So let's close this meeting. So in short, if I say, so today was a very productive session, and so thank you very much, Dr. Ravi Singh, Professor Parsad, and Dr. Juliana, for your time, for the information and presentations. So thank you very much. So nice.
Thank you. Thank you very much. It was
Thank you.
pleasure talking to so many people.
Yeah, there was 200 plus participants who were beneficiary from.
Yeah, as Dr. Norman Borlaug was used to say, you know, if something goes wrong in South Asia, you know, so that is going to affect the rest of the world. So we need to ensure that we all work together and do every bit which we could do to keep moving the productivity, no matter what environment we are going to face, because we are so many people there living, and we have to apply all methods, all approaches, policies to ensure that productivity goes up, and farmers should get their good price also, you know. I always find that is the most important. If you put good price for wheat, the yield starts to go up because farmers do manage their fields better.
Yes,
Thanks.
yes, exactly. So,
and I think if I could make one comment, I think it's important to take a systems approach. We need to use genetics in combination with agronomic. I think that's very critical. Only genetics or varieties may not solve the issue, so we have to really match the environment with genotype, and also consider the social and economic issues relevant to that. And also, I think the final point is we need to make sure that any technologies, whether it is genetic technologies or agronomic technologies, the most important aspect is the adoption and scaling. So we really need to identify what are the barriers for adoption and try to remove those barriers through appropriate policies. And also those of us who are working more in the research side should be able to, or we should identify the appropriate scaling pathways and the scaling partners, because as research organization, we are not good at scaling or making sure that our varieties or our technologies are adopted by the farmers. We really need to identify appropriate scaling partners so that we have an impact at scale, which will positively influence food and nutritional security. Thank you so much.
Yeah. Thank you. Take one hand raised at the 11th hour. Let me see who is. SN, are you hearing? Will you enable one person?
I have allowed his mic.
Yeah.
Ali there. If you have any question, you can ask, please.
Ali Uzair, you can ask question. Unmute yourself.
I think he cannot press, and we should move on to conclude.
Okay.
You can write the question and send by email.
Yeah.
Yes.
Yeah,
for sure. We will also put the recording of these lectures on YouTube and on our social media pages. And for sure, I will share your email addresses with the participants. So if they have a follow-up question, they can contact you later as well.
Yeah. Thank you very much, respected speakers. for your time. We are closing. Thank you very much.
Thank you.
Thank
Bye-bye.
you.
Bye bye.
Bye.
Bye.